This video will cover the anatomy of smooth muscle. Smooth muscle is more primitive than cardiac or skeletal muscle. It lacks visible striations, meaning the sarcomere relationship of myosin to actin is absent. However, it is as strong as skeletal muscle and highly resistant to fatigue. Smooth muscle fibers are smaller than skeletal muscle and are usually involuntary. They lack apparent myofibrils and cross striations under the light microscope. Fibers are thickest in the middle with tapered ends and have a single, centrally located oval nucleus. This slide visually represents smooth muscle anatomy. The sarcoplasm contains thick and thin filaments in a disorderly pattern, with 10 to 20 times more thin filaments than thick ones. Dense bodies, similar in function to Z-discs, are dispersed throughout the sarcoplasm or attached to the sarcolemma. Thin filaments stretch from one dense body to another. This slide provides a visual representation of smooth muscle structure. Contraction is generated by a sliding filament mechanism involving thick and thin filaments. This transmits tension to the thin filaments. Smooth muscle does not contain the troponin complex. Actin and myosin pull on the dense bodies attached to the sarcolemma, causing the fiber to shorten lengthwise and thicken in the middle. This slide illustrates the generation of smooth muscle contraction. Shortening causes a bubble-like expansion of the sarcolemma. The shortening motion is corkscrew-like, as the fiber twists in a helix as it shortens and rotates in the opposite direction as it lengthens. There are two main types of smooth muscle: single unit and multi-unit. Smooth muscle is much smaller than skeletal or cardiac muscle. It is organized into bundles or sheets and responds to various stimuli such as nerves, hormones, and mechanical stretch. Smooth muscle is innervated by the autonomic nervous system, with swellings along the axon serving as synapses. The firing rate determines neurotransmitter concentration and the physiological response. Smooth muscle controls organ size, substance release, and the rate of flow in various systems. Single-unit smooth muscle involves large aggregates of cells acting as one unit. Multi-unit smooth muscle consists of single smooth muscle cells, usually with a single nerve connection. Examples of multi-unit smooth muscle include the erector pili muscles, vas deferens, and the iris of the eye. Multi-unit smooth muscle fibers are independent, each with its own motor neuron terminal and few gap junctions, making control mostly nervous. They are covered by a thin layer of basement membrane that helps insulate the fiber. In multi-unit smooth muscle, an action potential stimulates only one single muscle fiber, and they seldom exhibit spontaneous contractions. They are found in the walls of large arteries, airways of the lungs, erector pili muscles, and the iris of the eye. Single-unit smooth muscle acts as a single unit and typically lines hollow organs such as blood vessels or the gastrointestinal tract. Single-unit or unitary smooth muscle is the most common type. It forms sheets or bundles of fibers that act as a single unit. Cell membranes adhere at multiple points, and gap junctions join the cells, providing electrical continuity. Single-unit smooth muscle forms part of the walls of large and small arteries and veins, as well as hollow viscera like the stomach, intestines, uterus, and urinary bladder. It forms large networks, and an action potential causes the contraction of the entire organ. The chemical basis for smooth muscle contraction involves actin and myosin filaments, similar to skeletal muscle but without the troponin complex. Contraction is activated by calcium influx, and ATP provides the necessary energy. Major differences between smooth and skeletal muscle include the physical organization, excitation-contraction coupling, calcium control, duration of contraction, and energy requirements. Smooth muscle is not striated. Dense bodies serve as attachment points for actin fibers, some connecting to the cell membrane. There are few myosin fibers within the actin fibers, with a 15:1 ratio of actin to myosin. This slide displays a visual representation related to the contractile mechanism. Smooth muscle contraction starts slower and lasts longer than striated muscle, potentially for hours or days. It can shorten and stretch more than striated muscle. And contraction is initiated by calcium influx into the sarcoplasm from outside the cell. The sarcoplasmic reticulum is sparse in smooth muscle, and calcium flows in from the extracellular fluid. The lack of T-tubules means calcium movement is slow. Smooth muscle tone is due to this slow calcium movement. It also has less ATP ACE activity, resulting in less ATP degradation. Smooth muscle requires significantly less energy than skeletal muscle. Only 1 ATP is needed per contraction cycle, regardless of duration. It reaches full contraction about half a second after stimulation, with maximum contractile force achieved within 1 to 2 seconds. The rate of contraction is 30 times slower than skeletal muscle, and contractions can last from 0.2 to 30 seconds. Smooth muscle contraction force can be about twice that of skeletal muscle, and it can shorten greatly, reducing the lumen of organs almost to zero. The latch mechanism allows for prolonged holding in smooth muscle. Once contraction starts, less stimulus and energy are needed to maintain it, conserving energy. It can maintain tonic contractions for hours with minimal energy or excitatory signals. The latch mechanism involves lower enzyme activation, allowing the myosin head to remain attached to actin for long periods. The stress relaxation response is another important characteristic of visceral smooth muscle. The stress relaxation response is the ability of the muscle to return to nearly its original force of contraction shortly after being elongated or shortened. Initial stretching causes contraction and increased tension, known as the myogenic response. Smooth muscle fibers can stretch while maintaining contractile function. Smooth muscle can undergo significant changes in length and still contract effectively. This response allows vessels and hollow organs to change size while maintaining constant pressure, which is likely related to the quote latch mechanism. Intracellular fluid calcium is the initiating event for smooth muscle contraction. Increased calcium influx can be caused by nerve stimulation, hormones, chemical changes, or mechanical stretch of the fiber. Smooth muscle lacks troponin but has calmodulin, a regulatory protein similar to troponin. Calmodulin combines with 4 calcium ions to become activated. The activated calmodulin calcium complex activates myosin light chain kinase. This slide displays a visual representation of the contractile mechanism. Activated myosin light chain kinase phosphorylates the myosin light chains using ATP. The myosin heads then engage actin, and cross-bridge cycling proceeds as in skeletal muscle. Contraction ceases when calcium levels drop below a critical level, and myosin phosphatase removes the phosphate from the myosin light chains. This slide displays a visual representation of the contractile mechanism. Myosin light chain kinase adds a phosphate group to the myosin light chain dependent on calcium. Myosin light chain phosphatase continually removes this group, deactivating the myosin head. Smooth muscle responds to stimulation from nerves, hormones, mechanical manipulation, and self-stimulation, known as automaticity. This slide displays a visual representation of the neuromuscular junction in smooth muscle. Acetylcholine, ACE, and norepinephrine are never secreted by the same nerve fiber. AUR can be excitatory or inhibitory, determined by the receptor type on the target cell. AUR and norepinephrine usually cause opposite reactions at a target cell. We now examine membrane potential and action potentials in unitary smooth muscle, which includes slow waves and spike potentials. In unitary smooth muscle, the resting membrane potential is variable, usually between -50 to -60 millivolts. This variable resting membrane potential is called the basic electrical rhythm, or BER, sometimes referred to as slow waves. This slide visually summarizes the stimulation of unitary smooth muscle. Slow waves are not action potentials. They are local, unstable resting membrane potentials that determine the rhythmicity of smooth muscle contractions. When the BER exceeds the threshold of about -35 millivolts, slow waves can initiate true action potentials called spike potentials. Spike potentials cause rhythmic contractions of smooth muscle. Increasing the number of spike potentials increases the force of smooth muscle contraction. Regulation by mechanical stretch, hormones, and acetylcholine causes the membrane to become less negative or hypopolarize the cells. This slide visually summarizes the stimulation of unitary smooth muscle.